Let's start, Kaushik.
Yes. Hello all, a very good afternoon. I am Kaushik, welcoming you all for the afternoon session of Vigyan Yatra 2020. I hope all of you are having a very thought-provoking time. In the morning session, we understood a lot about how zebrafish and Drosophila can be used to understand very basic science questions, though they are very small. In this session, we are going to learn a lot more, and I'm very sure about it. So, without any further delay, we are going to start the session. Our first session is going to be given by Dr. Pavithra Chavali. Work in Dr. Pavitra's lab focuses on understanding how certain genes involved during early development are exploited in cancers. For this, she uses cancer cells and mini brains as a tool to dissect molecular mechanisms common and different between neurodevelopment and in some brain cancers. Today, she is going to narrate how mini organs that are made in petri dishes in lab and can be used to understand basic human biology. With this, we are going to start the session. You can start, ma'am.
Yeah, thank you, Kaushik. Hi, all. Hope you are back from lunch and you are all now full of energy to listen to what we have to say. So today, my talk is going to be on a new revolutionary technology which has started probably in the last 7-8 years about how you can culture organ-like structures outside your body. Okay, we call them as organoids, and we're going to see how different organoids can give us an idea about how normal human development occurs and how we can address it to understand how diseases are manifested. Okay. So the pressing question is, what are mini organs and why do we need them, right? We have major organs. Why do we need these mini organs? We have a lot of model systems. So let's go back to evolution, okay? If we think about evolution, we all started, all the organisms today living on the earth have started probably from a speck. And we have, through the evolutionary process, we have seen fishes, amphibia, we've seen the reptiles, we've seen the apes, and finally, we have become humans. So in all this process, of course, there would be certain conserved mechanisms in our body. And there would also be something which is very different, which is why we have moved on from being a chimpanzee or a Neanderthal to being a human today. So while we have conserved mechanisms, we also have different mechanisms. So as a human, we are also very susceptible to a lot of stuff. Today, what we know a lot about how our body functions is primarily by using animals as our model system. The most well-studied organism, for instance, to decipher human biology is a mice, okay? We use mouse frequently for doing our experiments. If you look at the major internal organisms, internal organs like the heart, the liver, the intestine, the stomach, everything is common, right? So when we go for blood drug discovery, typically we start in the labs doing it in cell lines, simple cell lines, and then we translate these into a small animal model like a mouse or a guinea pig, and then we move on to humans, which is the clinical trial. So this part forms the preclinical trial where we could use animal models, and in phase 1 we move on into the humans. Okay, so you can see that the shape of this whole funnel is actually pretty narrow. We start from around 100,000 or 1 lakh compounds, and in the end, after 10 to 15 years, this whole process, we only end up with one drug. So imagine that you just walk up to the clinic and you get a paracetamol. To get you that paracetamol, we have taken for 10 to 15 years, and in the end, you have one paracetamol drug which is available. So like that, it is. If you think about this for paracetamol, you can imagine for more complicated drugs like stuff which is related to heart or brain, how much more we have to spend, okay? One, economically, it is not feasible because for one drug, you wait for 20 years. And two, the reason why most of these are failing is because of inherent differences between what we have in animal models and what we have as humans, right? And also because humans are unique, we have evolved specialized cells. So I'll just give you a few examples. So this is an example of a mouse versus a human brain. So you can see that this is not to scale, but you can imagine that for the size of mouse, the brain is really small. But what is even more striking is the human brain here has multiple gyri and sulcus. Okay, so these kind of folds that we see calling encephalization is not present in the mouse. So a lot of unique pathologies are present in humans. Okay, now if you think of any other organ system, for instance, if you take a kidney, you can see that to scale how big a human kidney is compared to a mouse kidney. Similarly, when you think of a mouse liver versus a human liver, or even if you take mouse intestine, you can see that there are subtle differences between the two. The gross morphology is the same, but there are subtle differences. And I think that we also should think about how our systems would have evolved. Okay, for instance, a mouse when it has fever is not going to have a paracetamol. When it has pain, it's not going to go and have a painkiller. Whereas as humans, we have all that, right? The intake is completely different. So your liver would have evolved to kind of metabolize those toxic substances. Similarly, when we think about our intestine, a mouse when it's hungry does not go and have a pizza or a burger, whereas the first thing we do is dial for a pizza or a burger. So the way our intestines work, the way our liver works, the way our major internal organs work is going to be completely different from when we look at other organisms that we use in the lab. Okay, and apart from what is there in our body, we are also susceptible to certain unique challenges. So let's take this very relevant example of COVID, right? So SARS-CoV, when you infect them with the COVID strain, these lab mice, they normally do not get infected unless they have humanized it, which means that you introduce certain human genes into the mouse to make it susceptible to SARS-2. Similarly, when they used macaque monkeys for checking these infections, they found that most of these macaques just had a mild infection and they were able to quickly clear it off their system. And we know that in the last 6 to 8 months, how much we are suffering, how much careful we have to be to prevent these severe effects because of COVID, right? It can even be lethal in case of humans. The other thing is, apart from these pathogenic infections, we also think about our genetic makeup. Okay, so there is a certain disease called disorder called as microcephaly, which in the morning Sonal and Megha would have spoken to you about. So certain genetic mutations in humans can result in babies with small brain phenotype being formed. Okay, they are called as microcephalic children. And these same genes, when they are mutated in mice, do not cause microcephaly. In fact, they result in dwarfism. So you can see that here it is not only the head, the whole animal is really tiny here. So these are certain genetic differences in the way our body is made up that results in these differences. Therefore, we have to think of something which is very close to human system if we want to come up with a very efficient drug delivery, drug discovery, and also to understand in general how our body functions. So the crux is that if we can mimic human organs, okay, and we have a better understanding of biology, we can address a lot of health issues efficiently. So when I say we can mimic human organs, you can't take a human and you can't perform experiment on a human, right? So the way to do it is basically what I've shown here. So we have to rely on our basics. Just like you can see a plant here which grows from a single seed, all of us have grown from a single embryo, okay, from a single cell stage. You've seen in cell division lecture by Megha that from a single cell stage, we have gone on to become the fetus, and then slowly the fetus develops over a period of 36 to 38 weeks, and we have an adult human, okay? And even when you are in your adult stage, your body is made up of almost 37 trillion cells. Okay, which means that we have multiple different types of cells. We have a heterogeneous composition. For instance, you have your brain cells, your blood cells, your liver cells, your muscle cells, and the stomach cells. All these are there in the human body. But each of them can come from a single stem cell population. So these stem cells are your seeds from which we can make different types of cells. But how do we make it? What we have to do is we have to add to these stem cells specific type of chemicals, specific type of growth factors, which lets them grow. So if you take a stem cell and you add one particular nerve growth factor, that is going to give rise to a nerve cell. So similarly, if you add something which triggers the blood a differentiation, you're going to make blood cells. So like that, we can use different cocktail of substances which will help us make different parts of our body, different organs in our body. Okay, so what we are going to see now is how certain specific mini organs can be made and how we can use them. So I've just highlighted here some of the mini organs which have been made so far. So for instance, from brain, we can make mini brains, which is the cerebral organoid. So we call these mini organs as mini organoids. Okay, they are organ-like, so we call them organoid. So people have made lung organoids, stomach organoids, kidney organoids, gut organoids, liver organoids, thyroids, and inner ear, and people have even made eyes. So in our lab, we are interested in making mini brains in the lab. So for this, I just have to quickly tell you about how our brain develops. So when we have 3 germ layers, the ectoderm, endoderm, and the mesoderm, from which all the other tissues grow, from the ectoderm, your skin and the brain develops, okay? And your ectoderm gives rise to this specific shape called as neural tube. A lot of you would have heard about it. This neural tube subsequently goes on to make brain and the spinal cord. So if we can succeed in making this neural tube and start making, adding certain compounds, we can slowly get it to this stage where we can make mini brains in the lab, okay? So how do we do that? So we take stem cells from a healthy person, okay? And we induce them by adding specific chemicals to make neural tubes. We give them proper media, just like blood and cerebrospinal fluid and the placental fluid in your mother's womb takes care of development. Like that, in the lab, we use specific media which closely resembles those physiological fluids to give rise to these neural tube formation. Once we have this neural tube, now we have to keep it in a maturation phase so that they can start forming this kind of a brain structure. So what we do, we have them in this physiological fluid and we put them on shakers. Okay, now they'll be on a rotating platform. So each of those neural tubes has now given rise to these small tissues, tissue-like structures, which we call as organoids. And this is within 3 weeks of you starting from the stem cells. And once we have this, we let them to mature and they reach almost 4 millimeters in size. And you can clearly see that this is a brain organoid which is developing, okay? And you can clearly see this black dot here marks the site where your optic lobe would develop. And if you think about the size of this organoid, it is as big as a Drosophila, the fruit fly which Dr. Sonal was talking to you about. Now we can keep growing these cultures for almost 3 months or 4 months, and if you have proper aeration, you can study a lot of phenomena which happens in the brain. But do these actually resemble any of the brain structures that we actually see? So what we have to do is basically, so here you see a much mature, more mature brain organoid. So this is in day 60, and you can see here an optic lobe has clearly formed. And when you do a cross-section of these organoid, we're going to see if it resembles a brain or not. So curiously enough, when we do a cross-section of the organoid, you start seeing specialized regions. So for instance, this is the gyri that we were talking about, this kind of a folding pattern that you see. That is what you see here in the brain as well, in the organoid as well. And even within this folded structure of the gyri, you can specifically start seeing different neurons coming up. Okay, so we know that these mini brains are very much like normal brains in the sense that during early development, this is how your brain would look like. Okay, now we have made all these mini brains and we have a lot of them, but what can we use them to study? So what can mini brains tell us? So for instance, several groups have told that have used these mini brains as a very powerful tool to understand what is common between us and our closest possible relatives, like the chimpanzees. Okay, so here you can see on the left side, you can see a human brain organoid, whereas on the right side, you see a chimp brain organoid. So you can see that there are a lot of differences. For instance, in humans, we have much more neurons here compared to the chimpanzees. So such evolutionary differences will help us understand how we function as humans. Okay. And the most important thing is actually, we can start seeing how specific infectious agents affect the brain. So in 2015 onwards, there was a big global pandemic like COVID, which was called the Zika infection, which specifically caused small brain size in children. Okay, so in fetuses, it was causing small brain phenotype. People did not know how the Zika virus was specifically affecting the brain. So people used organoids as a very strong tool. So they showed that in control organoids, everything was developing fine. And this is the layer of early embryonic brain. Whereas when there is a Zika infection, this complete layer is affected. So you see here a beautiful structure, whereas here it has completely collapsed. And this structure is your stem cell structure, which gives rise to the whole brain. Now, if you don't have this layer, you can't have any more brain formation. Therefore, you had children with small brain phenotype. And another interesting thing which we can do with these organoids is decipher how our brain functions in the form of neural circuits, right? So for instance, if you see pizza, your mouth waters, you feel like having that. If you see a tiger, on the other hand, you would run the other way around. So differences in such kind of a behavior are because there are specific neural circuits in your brain which fire at different rates. So with the organoids, you can, by adding specific chemicals, see how different parts of the brain fire differently and why that leads to a particular type of a behavior. Okay. So these are really interesting things that we can do with cerebral organoids, which is the prime focus of our lab. But apart from human brains, people have also tried doing a lot of stuff, going straight from the brain to the stomach. We have a lot of literature on this now. So a lot of scientists have been working on creating artificial intestines because food biology or gastrology, as they call it, is like a big industry. Okay, so when there is a problem in the stomach, then of course the whole thing crashes, the whole system crashes, because that is where you derive your whole energy. So people have made organoids from different parts of the intestine. So for instance, this intestinal organoids look something like this. Okay, most of this pioneering work has been done by Professor Hans Cleaver and his lab. So where they have done different types of colon organoid, ileum organoid, all those organoids have been done. And if you take a cross section of the stomach region of these organoids, you can clearly see that there is a beautiful epithelial lining, and you can even start seeing specific types of red cells even within these stomach organoids. So what can you do with these organoids, basically? So even before that, I would like to say that there has been a wonderful advance which has recently been made, where normally when we think of organoids, we grow them without any support. But recently, a group has developed a unique chip structure where they can seed into these structures specific stem cells, and you can look at epithelial cell growing and forming an intestine over a 30-day period. Now, this whole structure behaves like a proper human intestine. Okay, so this is like a really fantastic advance which has been made. This has just been done like a couple of months ago from a group in Switzerland. So what can mini intestines tell us? So what they can tell us is how we are susceptible to a lot of microbes. For instance, when somebody has a salmonella infection, okay, or when you have a typhoid, you have a lot of diarrhea because your body is completely, your stomach lining is completely washed off and there is a huge infection. Okay, so you can see here that if this is a control organoid, intestinal organoid, which is perfect, when you have a salmonella infection, the whole structure collapses. So all your microvilli projections are completely lost, and therefore you can't have food absorption. And more recently, people showed that if you have a stomach organoid, specific cells in that stomach can be infected by COVID-2 virus, the COVID virus. So this is a bit scary, but then it also gives you an information beforehand saying that normally we say that, you know, it's okay, even if it's parboiled or half boiled, your stomach can take care of it. But in fact, for this virus, there is no clearance system yet from the gut. Okay. And another very interesting thing is we can identify which types of drugs can be used to cure different infections and diseases. For instance, somebody might have an ulcerative colitis or a stomach cancer. So we can add different types of drugs to these organoids, gut organoids, and find out how we can get rid of those particular types of lesions. And Similarly, when we talk about COVID, obviously you can't escape talking about mini lungs or the lung organoids. So if we look at the whole lung system, the airway system, we talk about your trachea, you talk about your lungs, and within the lungs, you have alveoli, which are the gas exchange chambers. And scientists have been successful in making these alveolar mini organoids. So when you do an overall 3D image, this is what it looks like. So each of this particular structure you see here would be something like the red color balloon-like structure you see on your right, okay? And with the mini lungs, we can identify how microbes can kind of, infect them, and how we can clear them very quickly by giving different drugs. And interestingly, in the paper on the left, they identified that during development, there is a specific set of cells which are formed, which can help us avoid late stage lung collapse. Okay, that is a very interesting insight which has been provided by that particular paper. So it's not only about how we can address how infectious agents infect us and how we can get rid of it. It can also be how we understand basic biology to move forward in controlling human health or in regulating human health. And just like lungs, you also can have kidneys, which are your ultrafiltration devices within your body. And in the kidneys, this is quite interesting study where they showed that normal kidneys, kidney organoids grow like this, but sometimes people have a genetic defect which results in kidneys forming cysts in them. Okay, and those cysts, actually, when the organoids form cysts, they are such fluid-filled cavities which can be identified. Now, you can either correct the genetic mutations using genome alteration by making certain gene edits, or you can treat certain drugs, and you can see whether this kind of a cyst formation can be reversed or not. And the most fascinating endocrine organ of all is the liver, because that is your master endocrine organ, which secretes a number of enzymes or hormones. So for instance, here, what they have shown is they can culture different parts of the liver, because liver has different lobes, and they show that they can culture different parts of the liver. And in fact, they can even make bile ducts separately. Okay, and bile can be secreted from this liver. So this is especially useful model when people study certain liver degenerative diseases. Okay, some people can have non-alcoholic fatty liver. So in such cases, how can we treat it? What is the biology of it? All that can be quickly understood by using these mini livers. So what we have seen so far with respect to organoids is that we can use it to understand basic biology. So we can understand how organs are put together, how organ systems are put together, all that we can understand using organoids. So once we understand that, we can use them to model a lot of diseases. For instance, when there is a pathogenic attack, different types of pathogenic attacks, we can understand that. Or when there is a cancer progression, we can take stem cells from the patients, okay, and we can make it into particular organoids and see which kind of drug would that patient be responsive to. And certain genetic diseases like the polycystic kidney disease that I spoke about, that can be studied using these organoids. And when we talk about disease modeling, we are not far off from talking about drug discovery. Okay, so major pitfall so far has been that we use animals which do not really closely mimic a human system. In such cases, if we use organoids, we can kind of shortlist the drugs which can quickly be used to model which one would be the most responsive one and which one would be the more useful one, efficient one in case of human trials. Okay. And in drug discovery, there is something which has recently been catching everybody's attention, which is personalized medicine. Like I said, no 5 fingers of yours are the same. Each of them is so different. Each of us has different set of genetic makeup. Okay, the genes would be the same, but the way it is turned on and off would be completely different. So in such cases, if we take stem cells from that patient and we identify how we make organoids and we add a particular drug to see which particular drug will be useful for that patient to cure a particular disease, that will be something like a tailor-made medicine, which is personalized medicine or precision medicine. So organoids, they are small, but they are very powerful. Okay, but is everything good? We always have to take it with a pinch of salt. Okay, so If you think about our human body, organoids are not the solution for everything, because when you think about the human body, all the organ systems we have are linked. They are not isolated. Like when you feel hungry, your brain actually signals that you have to eat. So this kind of a nervous system, there is like a metro rail connecting your whole body and different organ systems, which currently in the organoids we do not have. Okay, organoids, we still grow them in Petri plates as isolated model systems. So we can understand how they develop, how they grow, how they come up with challenges, but how do we overcome this problem? So there are ways to do that. People are trying to do that. So they take individual organoids and they try to add other cell systems, other immune systems into that particular organoid and see whether they can activate the genes accordingly. So that is something which we have to remember. And the most important thing is that we are the sum of our environment and genes. So genes are not everything, basically. So if all our genes are the ones which are making us up, then all of us would be like these clones, getting up at 9 o'clock in the morning and going to school or attending your online classes. But in fact, we are not. So each of us has a certain environmental, we react to certain environmental stimulus, which makes us go through a lot of these emotions, which in turn can actually switch on and off a lot of receptors in our brain, which in turn controls the whole body system. Therefore, while organoids are a powerful system, we still have a lot of way to go forward in making them into something which we can routinely use for our purposes, for the biology purpose. So with this, I would like to thank you and take any questions.
Thank you so much, Dr. Pavithra. That was a very wonderful talk. So we have a question from Mohammed Sohail. So I guess he is asking how we can trust. So as you told that these drugs can be used to treat organoids of the disease system. So how we can trust, how much we can trust from that the same drug can be used to treat the same disease. I guess this is his question.
Okay, so there are multiple ways to do that. When I said that there are limitations for organoids, you can also think of other ways. For instance, instead of taking just a mice and putting the drug into the mice, we can take these organoids, we can implant these, so we can go for xenograft models. So these humanized organoids can be put into a mouse model, and then you can see how things are going. That is one way which people are trying to address. So they try to make humanized mice model based on grafting these human organoids into the system, and we can study that. So that is one way of circumventing it. And you never do one organoid or two organoids, okay? You go for a whole plate, you do it in multiple batches, you look at multiple animals. So that is how you have to do it.
So I'm having a question in this topic. So since as you have already answered it, but as you told, these organoids are isolated organs, let's say. But if I want to study a whole systemic model, how these organoids, is it possible to combine organoids in such a way so that I can study whole system? Since you have showed the small intestine formation, so is it possible, are we seeing any future direction in that way?
Yes, actually, specifically for the intestines, people are trying to put together different intestinal model, like the colon organoids, the gut organoids, everything. People are trying to put it together and make it into a single system and see whether it can work or not. So even in the case of brain, what we have been thinking of is basically we make forebrain, midbrain, and hindbrain, because these are really tiny, right? So we try to culture them together. And inherently, they have the capacity to fuse and form a whole brain-like structure, which is really interesting. So the self-organization of these structures is what makes them very powerful.
Yes. So I guess that's all I guess we are not seeing any questions. Okay. So thank you so much, Dr. Pavithra. So now we are going